Okay, hopefully everyone got enough sleep last night, so everyone's wide awake for a lecture today. Maybe it's a good thing, especially this one is screencast, because you guys I might fall asleep in the middle of this. I sometimes think if I wasn't talking, I would put myself to sleep. Hey, I should try listening to my screencasts before I go to bed. It might help. Okay, so we left off talking about, or we just finished talking about the lytic cycle that lambda goes through. So in order for lambda going through the lytic cycle to package its genome properly, it needs to switch into this rolling circle replication in order to generate linear forms of the genome. And so these are going to be packaged into virus particles, and then the bacterium is going to be sacrificed to the viral gods and will be released all kinds of little viruses from that cell. But now let's talk about the more interesting option that lambda and other temperate bacteriophage have. They can enter into this cooperative process or arrangement with their host cells called lysogeny. So we talked a little bit about the conditions that mediate or favor lysogeny over lysis. One of them is shown here in this figure that I took from your textbook, and they only talk about one of them, and that's this high multiplicity of infection. But also remember that the other, and I'll talk a little bit more about why high multiplicity of infection or how high multiplicity of infection mediates lysogeny versus lysis. But also remember that this same process is going to occur, and I added this part, under nutritionally depleted conditions. So two different conditions stimulate lysogeny when lambda infects E. coli. And so what's shown here is that region where I told you that we would be spending a lot of time talking about. This is the control region of the lambda genome. So here is the promoter right, here's the promoter left, the CRO gene. I'm sure these are all just fresh in your mind from Monday's lecture. But I also mentioned that you can always remember the genes that are important in the lysogenic lifestyle because they always have conveniently the prefix of lowercase c. So at the gene level, bacterial genes or viral genes are usually italicized and the first letter is lowercase. So when you're talking about, for instance, this gene here, C2, its gene product would be represented here, having a hard time seeing this morning, as uppercase C2. But when you see the gene or the gene product of either C1, C2, or C3, those are the genes whose products are going to functionally drive the system towards lysogeny and keep the cell from going into lysis. So when a bacteriophage lambda infects at high multiplicity of infection or a nutritionally depleted E. coli cell, the conditions are such that these two genes, which we didn't talk about on Monday, C2 and C3, are not only transcribed and translated, they persist. This is a key point. C2 and C3 to some degree are kind of wimpy little proteins. Think of them as these ephemeral little proteins that live in fear of a proteolytic enzyme goes by either of two names, FTSH or HFLA. This is a normal E. coli protease that's involved in autophagy within the E. coli cell. So it breaks down proteins to recycle the amino acids to be used again. So as proteins approach the end of their life cycle, there are proteases that help the cell recoup some of the investment that it's made in that protein. And so C2 and C3 just happen to be very, very good templates for these enzymes. Now, the reason that high multiplicity of infection favors lysogeny is because at a high multiplicity of infection, that one E. coli cell has probably had 10, 15 or so lambda genomes injected simultaneously at one time. When that happens, the transcription of the genes C2 and C3 and then the subsequent translation into the protein product can literally overwhelm these proteolytic processes. There's so much of these ephemeral little wimpy proteins made from all of these genomes that came in at the same time, this protease just can't chew them up fast enough. And so it's the persistence of C2 and C3, and they actually cooperate with one another. So C2 and C3 actually bind with one another and kind of protect each other, look out for each other from this big mean protease that's trying to chew them up. So you can have persistence of C2 and C3 under high multiplicity of infection. You can also have the persistence of C2 and C3 when the bacterium is nutritionally depleted. This protein, FTSH, or whether you call it HFLA, which I think is the older name for it, is actually an enzyme that is, first of all, depleted to some degree in a nutritionally challenged cell. Cell can't make much protein, so it doesn't produce this protein very much. But probably more importantly, this FTSH protease is negatively controlled by cyclic AMP. So remember that when an E. coli cell is not doing very well, if it's not importing glucose, its adenyl cyclase is activated, and so cyclic AMP levels are high. We talked about cyclic AMP as binding to the CAP protein, the catabolite activating protein, and then it activated that protein. The opposite is also true in some cases, and this is one of those cases. So when the levels of cyclic AMP are high, they bind to this protein and reduce the activity. So a protein that is, or a bacterium that's not doing very well nutritionally has low activity of this protease. So that's a second condition that you can see that promotes the production of C2 and C3. And so this is the setup. This is the initial stage which is necessary for the virus to make the decision to go into lysogeny rather than lysis. If the cell is happy and got lots of nutrients and it's only infected by one virus, C2 gets chewed up just as fast as it's made and the system is pushed towards lysis. But now imagine that we've infected a cell that's struggling. And so C2 and C3 persist for a little while. So what happens with this C2 protein is it has the ability to bind as an activator. The C2 protein has the ability, where is it on here, to bind to, it's shown down here. Actually, it's not shown on here. It actually has the ability to bind as an activator for C1. So if C2 persists, one of the first things that it does is to promote the transcription of C1. C1 is the protein that's battling with CRO. So I mentioned that what really the battle between lysis and lysogeny comes down to who wins the battle of these two proteins. If CRO predominates, lysis occurs. If C1 predominates, lysogeny is going to occur. So the C2 protein actually binds and activates this promoter here called, I don't like the definition that this, or the abbreviation that this book uses for this. They call it P sub RE. I prefer that it gets confusing because P sub R means a rightward promoter, yet it's transcribing to the left. So in other slides, I'm going to show you another set of slides which may make this more clear, or it may muddy the waters. I'm not sure which. You'll see that this promoter is, I think, more frequently and more appropriately just called P sub E. E stands for establishment. This is the promoter that's necessary to establish lysogeny. C2 acts as an activator for this protein. An activator for this promoter, which drives the transcription of this C1 protein. Now, this C1 protein is the lambda repressor. So make a mental note of the fact, or a paper note if you care to. We use the terms C1 and lambda repressor interchangeably. They are the same protein. Two different names for the same protein. The lambda repressor, or C1, is going to bind to that same operator site that CRO bound to. So we talked on Monday about the fact that the CRO protein, if it's made during the initial part of the infection, is first going to bind to operator right site number 3. That's the highest affinity for CRO. And when CRO binds there, it serves as a repressor so that neither of these promoters that are pointing towards C1 can get past that repressor. CRO is doing its best to try and stop the transcription of C1. But now we're talking about a situation where C1 is gaining an upper hand. It's the protein that's produced first, or maybe a little bit more is produced early on. What happens with that C1 protein is it binds to the exact same 3 sites that CRO does, but it does it in the opposite order. So if CRO has as its highest affinity operator right section 3, and its lowest affinity operator right section 1, it's just the opposite now with the lambda repressor, or C1. It binds first to C1, then if it builds up to higher levels, it binds to C2, and if it binds up to still higher levels, it binds to site number 3. But if you look at this, you can see that when site number 1 of this operator is bound, the first thing that that's going to do is to stop the transcription of CRO. So these two are really in a battle to stop the transcription of the other gene. CRO binds to site 3 to stop C1 transcription. C1 binds to site 1 first to stop CRO transcription, to push the system a little further, to make it a little bit more dedicated to the lysogeny pathway. But if you look at where this C1 binds in this operator, remember that the transcription that C2 was driving is from this promoter for the establishment of lysogeny, P sub E, or in this cartoon, P sub RE. If you stick proteins onto this operator, whether it's CRO or whether it's C1, RNA polymerase doesn't get past it. So what happens now is that you've begun the transcription of C1 from this promoter for the establishment of lysogeny, and then quickly you shut it off. Lambda shuts its own promoter off by binding to that site 1. But this is where the lambda repressor is actually kind of a misnomer. You could actually call it the lambda repressor / activator. Because while C1 bound to this site number 1 and site number 2 are repressors for CRO transcription, they are simultaneously activators for this other left-hand oriented promoter called PRM, or in subsequent slides, and I prefer it as P sub M. M stands for maintenance. This is the promoter that's dedicated to maintaining lysogeny because it maintains transcription of C1. So C1 doesn't shut itself down, or it shuts itself down for one promoter, but it activates another promoter. So it binds next as C1 gets to higher levels. It binds to site number 2, which either continues as an activator or has no additional effect. But then if C1, this lambda repressor, binds or rises to even higher levels, it's going to bind to site 3, where it's actually going to shut itself down. This is autoregulation. So the lambda repressor actually regulates itself once it's established so that it neither rises too high nor falls too low. And we'll talk about why it's important that this lambda repressor never rise to too high a level. So this is actually it's a little bit of a busy slide, but if you go through it, hopefully, the entrance to lysogeny pathway becomes a little more apparent. Or perhaps did you have a question, BR? Yes. So CRO has its highest affinity for the operator right section 3, which is just the opposite of C1. So so no new information on this slide, just an alternative way of looking at it from an older textbook that I'm still somewhat fond of. So here is that same operator site. Here you have the C1 gene oriented towards the left. Here's the CRO gene oriented towards the right. And these salmon-colored arrows contained within the DNA indicate the direction of these promoters. In other words, the orientation that they would drive RNA polymerase. So here is my preferred abbreviation, P sub E, for the establishment of lysogeny. So when this C2 activator protein builds up, and it can only do that, remember, at high multiplicity of infection or nutritionally challenged E. coli. But whichever one of those conditions allows the protein C2 to bind to a higher level, it's going to bind adjacent to this P sub E and act as an activator. So C2 is an activator protein for the promoter for the establishment of lysogeny. So this is going to drive transcription to the left, and so this is where you get the first whispers of transcription of the C1, the lambda repressor. Those transcripts will then be translated into protein, so you'll get a small amount of this C1 protein produced. So, yeah, so C2 is normally broken down under healthy conditions by a protease called HFLA, and I've already, of course, I've gotten ahead of my bullets, but it's the abundance and activity of that protease, which actually I think goes by two different names, that dictates whether C2 and C3 build up. So under starvation conditions, that protease level is low, but the HFLA is high under nutritionally replete or healthy conditions. So there's two reasons for that, is that under stressful conditions, the bacterium is simply not able to synthesize as much of that protein, so less transcription and less translation. But probably more importantly is the effect of cyclic AMP as an allosteric modifier. So cyclic AMP levels are high, they bind to the protease is less active, and so C2 levels increase. So C3 is a stabilizing protein that helps to protect C2 from that protease to give it a chance for lysogeny to occur. So, but C2 begins the transcription of C1. The C1 protein is now going to bind first to site number 1. And I like this slide because it shows, and I'm going to show it in yet another slide, but I like this slide because it shows that site number 1, where C1, or the lambda repressor, is going to bind first, is pretty much right on top of promoter right. Promoter right was the first promoter that we talked about. This is the promoter that drives the transcription of CRO, and it makes all three of those different types of the rightward transcript through O and P and sometimes through Q. But the first thing that the lambda repressor does is to stop, as best it can, any transcription of the CRO protein, because CRO would push towards lysis. But you'll notice that, again, as I said in the last slide, if you put a repressor protein bound to this operator site, it's going to stop RNA polymerase coming from the promoter for the establishment of lysogeny. It's going to block it. So as soon as C1 transcription has begun, it almost immediately stops. But now, by binding to site 1 and to site 2, those same repressor proteins now activate the promoter for the maintenance of lysogeny. So you now secondarily get transcription of C1 from a different promoter. So now the system is now almost perhaps irrevocably committed towards lysogeny, because now it's pretty much for sure shut down CRO and is producing probably even more of it, of the C1 protein. So this cell is, or this virus and this cell are probably pretty much destined to enter into a kind of a mutualism. Well, I guess you could, I think of it more as a ticking time bomb, where the virus is going to insert its genome into the host cell as a prophage and turn the E. coli cell into a lysogen. So the lambda repressor, or C1, I should have that in capital because now I'm talking about the protein, like CRO, acts as a repressor. For promoter, we're talking about promoter right, but the same process is going on from promoter left, the promoter going in the other direction. But it binds to these sites just in different order. So again, I'm being a little bit redundant, maybe a lot redundant. It's binding first to site 1 to shut down CRO. Yes, and again, I'm just repeating myself because these are important points, and the more I repeat them, the more easily you'll do on the exam. Yes, question? Mm-hmm. So when it binds to it, so the question is how does it actually get more of itself? So if you stop the transcription by binding to site one, so you're not getting transcription from promoter for the establishment of lysogeny, binding to site one and perhaps also to site two, but certainly to site one activates this other promoter, the P sub M for maintenance. So you switch, you're constantly producing transcripts for C1, you're just doing it from a different promoter slightly later. And remember, the entire process here is about 30 minutes in length. So these decisions are being made very fast within these cells. The time that I'm doing this lecture, several cells are probably going into lysogeny. And again, this is just repeating myself that C1 and Crow bind to the same sites, but with different affinities, so opposite order. So am I also repeating myself here? So the lambda repressor binding to site 1 shuts down promoter right, hence no Crow protein. It also acts as an activator for the promoter of maintenance. So yes, so I've, again, as is typical for me, I've gotten ahead of my, my bullets. But an important point that I want to bring up before, because we're talking about lysogeny, is this process of autoregulation. So as C1 levels rise to a certain level, eventually they bind to the lowest affinity site, number 3, and you can see that that binding site is on top of promoter for maintenance. That will stop all transcription for a moment. So what happens is that now C1 is no longer transcribed at a high level. C1 proteins eventually will drop. The protein, the first one of these 3 sites that C1 protein is going to fall off of is the lowest affinity site, site 3, and that's going to allow transcription. So what this allows the lysogenic bacterium to do and the prophage is to maintain a homeostatic level of C1 that doesn't rise too high. And we'll talk about why it shouldn't rise too high. And again, I love lambda. Students don't love lambda. So what I try to do is anytime when I'm scouring the internet, doing Google searches for lambda genetics, anytime I find a figure that I think makes some sense and perhaps could potentially help you guys, I just put it up. So it's, again, maybe this one works a little bit better for you because although it should C1, so this slide has a problem that I should adjust. It makes it look as if the first gene downstream from this promoter for the maintenance is the N protein. N is actually further downstream. There should be the gene for C1 to the right of that N gene. But again, you can see that these lambda repressors are binding, and now you actually see maybe an anatomically better representation of the binding. Because these lambda repressors, like most DNA binding proteins, bind as homodimers. And they're binding to inverted repeats. So what you're not seeing too well here is shown down in this salmon pink color box here. This is what the operator sequence is. And remember that operators, because they bind DNA proteins, oftentimes have inverted repeats in them. So that's what these arrows pointing towards one another indicate. And so each one of those arrows would bind one C1 protein or one Crow protein, same thing, just different affinities. And so there's actually 2 proteins that bind to each one of these sites, and the proteins interact with one another. So non-covalently, but this stabilizes this interaction of this repressor / activator. So it's repressing Crow and it's activating C1. Also when it's bound here to this site 2, this little blowout here is meant to indicate that it's serving as an activator for RNA polymerase. So RNA polymerase now binds to that promoter and transcription occurs. So hopefully one of these slides will really help you understand lambda, because once you understand lambda, you'll realize that it actually is really pretty cool. Once things just kind of fall into place and you realize that it's more than alphabet soup, because I know there's a lot of different genes and single letter names. They're hard to remember, but once you understand it, it's a really cool system. And yeah, I think I'm getting sick of this at this point. And I don't really know why I put in just another bigger version of one of my favorite lambda genomes. There was probably a point, but it escapes me right now. But we've now talked about the gene expression that's necessary for lysogeny of lambda. You need to stop the CRO production and produce the C1 production so that the lysis proteins are not produced. But there's another really critical thing that has to occur for a lysogen to be created. First of all, we have to first stop those late proteins from being expressed. So that's what the C1 protein did. It stopped CRO expression, and so it ends up actually stopping the Q expression, which ends up not allowing the expression of all of those lysis proteins and the viral structural proteins. So that has to occur in order for lysis to occur. And C1, the lambda repressor, is doing that. But the other really crucial thing that has to happen is that one of those lambda genomes has to be incorporated into the chromosome. It has to be subsumed into that chromosome. So that's what's shown here. And this is driven by a viral protein called integrase protein. So maybe if I go back, just for orientation, over on this part of the chromosome are the genes that are involved in recombination. You can kind of get a hint about what these genes do. INT, that is the integrase. So this is the protein who's going to drive the integration, the establishment of this prophage. Later on, we're going to call on the protein excisionase. And you can tell from that very appropriately named enzyme what it does. It's going to help to reverse the process. It's going to be able to rescue or resurrect that virus genome from within the lambda genome. So what's going to happen here is that the viral integrase protein is going to now cut both the viral genome, and it cuts it at only one particular place called the ACT site. We've already covered this, standing for attachment. And it also cuts E. coli in one and only one place. So E. coli can only incorporate one lambda genome most of the time, under normal conditions, because there's only one site in the E. coli genome that's recognized by this integrase gene. And so the bacterial attachment site, which is not labeled here, but we talked about this in specialized transduction, matches up to some degree with the attachment site in the lambda genome, and then that integrase is going to cut both strands of the viral genome, cut both strands at that one particular site in the E. coli genome, and then knit them together. So that now that viral genome has been subsumed within the larger circle of the E. coli chromosome, and now this is a prophage. And so this bacterium is a lysogen. So once it's incorporated as a prophage, how does it re-enter the lytic pathway? Because this virus has adapted this situation to avoid extinction. Because remember, it's gone into lysogeny probably because the E. coli cell might be going to die. It's found itself in a cell that doesn't have a lot of nutrients in it. So it puts itself in there, into the chromosome, and it waits. And hopefully the cell survives, and it finds itself in better conditions, and it's dividing. But at some frequency, the virus needs to get out of that chromosome, especially if the chromosome has any indication that the bacterium might die. So this is where the virus is just really evolutionarily very clever. And I mentioned this in Monday's class. But the virus has actually learned to eavesdrop on the conversation that E. coli has upon DNA damage. We talked about the SOS response system. So remember that in DNA damage, when the SOS response is induced, what happens is that REC-A detects single-stranded DNA. That's an abnormal signal. REC-A becomes activated. It becomes a coprotease to degrade LEX-A. C1, that lambda repressor, is actually very similar structurally to LEX-A. So this activated REC-A can't tell the difference in reality between LEX-A, which it wants to degrade to release all of those DNA repair mechanisms, or C1. So it's degrading both of them. what happens when you stop or degrade this lambda repressor is that now you've taken the governor off, right? So now all of these genes can be transcribed. So you can transcribe CRO, C2, OPQ. You can also express, very importantly from this other side, the excisionase. So under conditions that might cause the bacterium to die, the virus resurrects itself from the chromosome using its excisionase enzyme, circularizes itself, and then tries to go into the lytic cycle. This way, the virus has the possibility, at least, of lysing that bacterial cell and releasing 50, 100 virus particles that may actually survive and infect a healthy cell, as opposed to just dying stranded within the chromosome of that cell. Now, so I put a website on here, and I can't actually get it to work directly in my PowerPoint, so we'll actually just go to it, because it's got more detail than I really care for you to understand, but you should go through it, because it actually, it's pretty, I like it. It's pretty nice. So anyway, here you have lambda. So we're going to go through very quickly the entire life cycle, the lytic life cycle, not the lysogenic life cycle, but the lytic life cycle of lambda. So it binds first to that outer membrane protein LAMB, which is involved in allowing maltose across that outer membrane. Oh, click next. Then don't worry about the identity of many of these proteins, but then the lambda is going to inject its double-stranded DNA into the cytoplasm of that cell. Remember, it's linear when it's in the lambda, and it will circularize almost immediately upon introduction to the cytoplasm of that cell, based on these 12 base pair cohesive sites. So COS is an abbreviation for cohesive. These actually are, complementary bases. So once that base pairing has occurred, the host E. coli DNA ligase will make those phosphodiester bonds to make that a closed circular DNA circle, and then DNA gyrase from the bacterium again will help that DNA supercoil. I don't really honestly know what the function or the necessity of supercoiling is in this case, because the virus, if it's going through the lytic cycle, will first replicate itself by theta-type replication. So not rolling circle replication yet, but it will undergo this this theta-type replication, so that 2 circles become 4, 4 become, 2 become 4, 4 become 8. And then there's a lambda endonuclease that's going to help this virus initiate rolling circle replication. This should seem familiar to you, because this is very similar to a protein that you're going to be asked about in a question on the exam this week. Remember TRA-I, the relaxase protein that you're studying about when we talked about F plasmid conjugation? So the initiation, the generation of a NIC in the F plasmid that facilitates rolling circle replication, that allows conjugation to proceed, is very analogous. These proteins are completely different, but it's very analogous to the lambda endonuclease that's creating a NIC in one strand of this circular lambda genome, because that's what allows the bacterium to switch from rolling circle from theta-type replication to rolling circle replication. So one strand is cut, and then this can just roll with new DNA being synthesized here to drive around this circle. So it's just peeling off a strand. This is where it differs from the F plasmid in conjugation. In the F conjugation, this single strand DNA that's generated is passed via that type 4 secretion system into another cell. In this case, there's no other cell involved, and that single-stranded DNA, now lambda, not an F plasmid, becomes a template to synthesize the other strand within that same cell. In the F plasmid conjugation, the resynthesis of that strand occurs in the transconjugate in the other cell. So these are very similar processes, but they have their differences as well. So this is synthesized, but now rolling circle replication will continue, and these will just be replicated into what's known as a concatemer. So here at these COS sites is where these genomes are going to be cut up into bite-sized chunks that can be packaged into new baby viruses, new little lambdas that are going to be born from this cell at each one of these COS sites. But so they're formed as a concatemer. So you can imagine that if you have even just 4 lambda circles all going through rolling circle replication, you can generate a hell of a lot of genomes in a fairly short period of time. But now, how these genomes are packaged is really cool because, first of all, you have the expression of several of these late proteins whose identities we're not even going to go through because that would mean that I would have to learn exactly what they did. But these are the lambda proteins that are actually being put together into the capsid of the virus inside the cytoplasm, and it's being done by the bacterial's own chaperones. So this is like helping somebody rob your house. So GrowEL here and GrowES are chaperones that E. coli makes that are now being hijacked into putting together the viral capsid of lambda. So this will put together the head. Don't worry about the identity of these proteins. But what's really cool is that there's a protein so here's the pore through which the DNA is going to be put into this empty space inside this icosahedral head. That protein that's there is the terminase protein. That's the protein that actually has the ability to recognize those COS sites and to cut it. So it cuts it on both strands at this COS site, but remember, those COS sites had staggered ends. So it's not cutting them directly, 2 phosphodiester bonds directly across from one another. They're offset so that there are 12 base pair single-stranded overhangs every time this terminase cuts that COS site. But it does this as it's packaging it. So it cuts that into these asymmetric ends and then drives that linear DNA into the head until it hits another COS site. And then it stops. It cuts that COS site as well. So another head can begin to package this next member of the concatemer. So it's a very efficient process for packaging. So then there are I wish there was a T protein here because they put in the W protein and the F protein, and you should have a T protein there in the middle. It would at least make things easier to remember. Now, here's the other neat thing about how viruses are put together that's really different, is they're put together in an assembly line type of structure where, like in a car factory, you would put the engine together in one part of the factory and you would put the chassis together in another part of the factory, and then when they're both finished, you put them together so that you have a functional car. And so that's really what's going on here, is that the tail proteins are assembled here, or they actually self-assemble, and they're processed. And then once that tail is formed, it will actually now be attached, and only now be attached, to the icosahedral head, which already has a perfect lambda genome, linear in form, packaged within that icosahedral head. Now, here are those late proteins. The S protein is actually a protein that's referred to as the Holin protein, Holin. And that's a very appropriate name because it puts a hole in the membrane. That's where it actually gets its name. So the Holin protein, this S protein, which is one of those late proteins controlled by Q, pokes a hole in the cytoplasmic membrane of that bacterial cell. And the other protein that's encoded right along with that is the R protein. R protein is the lysozyme. That's the protein that degraded this little bit of peptidoglycan here to help these viruses escape. In reality, now with these holes poked in here by Holin, the cell probably is depolarized. It's probably going to lyse by osmotic pressure. So this next part of this animation I don't really agree with, but it illustrates it. I think the cell just lyses and bursts open and the cells are just free. I don't think they line up like this and pass out through that pore. But in either case, you get the point about this. So I really like that. I really like that website. So it will, hopefully help to make things clear for you. Okay, I think I probably already talked about this point enough too. So I mentioned that what's favoring lysogeny, oh, I probably should go back to slideshow. So these conditions of either nutritionally challenged cells or high multiplicity of infection drive lysogeny within the E. coli cell. And I think I've already talked enough about why the high multiplicity of infection drives lysogeny. But here it is in words again, so it's in your slide set for your studies. So it's actually the fact that these C2 and C3 proteins are produced in sufficient quantities, even though there's protease there, they just overwhelm it. So they can drive the system towards lysogeny. And again, I mentioned this already, but I want to make sure that people understand why, because I'll almost certainly ask a question on an exam like this. Why would it be advantageous for the virus to induce lysogeny under the conditions of high multiplicity of infection? Remember, it has to do with the fact that viruses, if they lyse every cell in their environment and they infect them all and lyse them all, now they're released into the environment with no one left to infect. They're just hanging around, waiting for a protease to break them down, waiting for an ultraviolet light ray to damage their DNA. And because they're viruses, because they have no repair mechanisms, they have no physiology, they're gone. They're now inactivated. So viruses don't like to actually kill all of their hosts. So if the condition is such that there are too many viruses for the hosts, it drives the system towards lysogeny so that the virus doesn't eat itself out of house and home. So it's a really clever evolutionary adaptation that these viruses have done. But really, why do we spend so much time talking about lambda, besides the fact that lambda is just one of the best known, best understood genetic systems in biology? And we've learned a lot about retroviruses and other viruses from studies of lambda. But there's actually more important reasons for that. And it's because of something known as phage conversion, or phage conversion, however you want to pronounce it. A lot of bacteria that are medically important, but also just environmentally important, actually have a different phenotype because they've been lysogenized by some type of a different phage. So E. coli lysogenized by lambda, so far as I know, doesn't have anything of a different phenotype. But there are some serious pathogens. Probably the best known example is the bacterium Corynebacterium diphtheriae. You can probably tell that this is a bacterium that causes the disease diphtheria. Fortunately, we don't see much diphtheria anymore. It's still responsible for killing about 3,000 people worldwide every year. You'll probably never see it in the US because each of you have been vaccinated. Remember the DPT vaccine that you've had? The D stands for diphtheria, in case you didn't know that. So you've been vaccinated against a toxin that this bacteria produces. But the gene for that diphtheria toxin, which is actually responsible for the sometimes lethal effect. So 5 to 10% of children who have diphtheria are going to die. And about 5 to 10% of people my age and older are also going to die from diphtheria. But this gene that encodes this lethal toxin is actually part of a bacteriophage. So diphtheria, Corynebacterium diphtheriae, which are not lysogenized by that particular bacteriophage, can never cause disease. They can colonize your pharynx just fine. As a matter of fact, they used to be part of our normal flora. But now, because vaccines, the DPT vaccine has been so effective, probably none of us in this room have strains of Corynebacterium diphtheriae in our throats because the vaccine has made us immune to even be colonized by these bacteria. But this disease, this lethal disease, is only due to phage conversion. The same thing with E. coli that causes hemolytic uremic syndrome, which is very lethal for small children. This is the hamburger strain 015787. It actually has the Shiga toxin, which is a gene that's found in a virus that normally infects Shigella. But now it's gotten itself into and lysogenized a certain strain of E. coli, which can now cause hemolytic uremic syndrome, especially in children. The same thing with streps and staphs. A lot of their virulence factors came to them accidentally by having a virus infect and become lysogenized within them. The cholera toxin, remember you're studying about cholera toxin for this next exam. That gene, which causes the entire pathogenicity, the entire virulence of that disease, is actually a gene on a virus. So part of the evolution of virulent cholera bacteria required a phage conversion, a lysogen form. So it's really, so this is part of the reason why it's important that we talk about lysogeny. And again, if you want to know more about this, or if my explanation isn't good enough, I put up another website for that you can look at. Okay, please don't applaud, but we are now done with bacterial viruses. No more lambda, I promise. We're now going to go into viruses that I think many of you are perhaps more interested in. So for the rest of the virus section of this class, what I'm going to do is to go by example through single-stranded RNA viruses of the positive sense, single-stranded RNA viruses of the negative sense, and then we're going to go into retroviruses. So we're going to go first through poliovirus as a system. Poliovirus, we're going to use that as a system because it's very well studied. It's nearly eradicated, sadly not completely eradicated, but it's still, not just for historic reasons, it's important that we understand poliovirus. When we're studying poliovirus, we're also actually studying rhinoviruses, so the cold viruses that probably some of you have right now. Hepatitis A virus, which hopefully none of you have right now. Foot and mouth disease virus, so those of you going on to study veterinary medicine so this system that you're going to learn about with the poliovirus actually applies to viruses that you'll deal with more readily. But it has such a great historic significance, and it's so well studied that we use polio. So polio, the disease itself, is actually more appropriately referred to as poliomyelitis. It's typically not the kind of disease that you might think it is. So you think of poliomyelitis, even though you've probably never, I hope, or perhaps never encountered anyone who survived polio. It doesn't always result, matter of fact, most times it doesn't result in the involvement of the central nervous system. So there's not always a paralysis or withered limbs that are associated with it. Typically, this virus is really highly evolved to simply cause a very mild, sometimes asymptomatic gastroenteritis. You get a little bit of diarrhea, perhaps, you don't really know why, maybe you get some shivers, a little bit of a fever, but maybe not even that. And then you recover and you're fine. You never knew what happened. That's actually what the virus wants to do. You know, the involvement of the central nervous system is a complete accident in terms of the virus. Infection of the central nervous system in a human and causing a polio is actually a dead end for this virus. The virus is spread via diarrhea and fecal water and contamination of water with that feces. It doesn't, if it gets into the central nervous system, it never gets out. So it doesn't help the bacterium to spread. So it normally infects only cells in your pharynx and enterocytes, the cells that line your intestinal tract. And unfortunately, just by random chance, that same receptor is also present on the anterior horn cells of the spinal cord. So if the virus gets access to the central nervous system, it has the capability to infect central nervous system cells, but it's purely accidental from the virus's point of view. It really just wants to infect the pharynx and cause diarrhea, increase its numbers, cause some diarrhea, and spread to many new naive hosts. It's actually perhaps the next viral disease that's going to be hopefully eliminated. Wild-type polio, and I put wild-type in parentheses because in many parts of the world, we still use a vaccine that's a live attenuated vaccine called the Sabin vaccine. Occasionally, that virus, even though it's attenuated, mutates back to the ability to cause disease. That's why we don't use that vaccine in the US anymore. If anyone in the US needs to be vaccinated for polio, for travel reasons, like military people going to Afghanistan where there's still cases of polio, they'll actually be vaccinated with a different virus so that there's no possibility that it can revert to a virulent form. So I'm gonna stop at this point because this is such a great story. I don't want to spoil it by trying to rush through it at the end. So we'll pick up with this on Friday. I'll see some of you tonight from 5:30 to 7:30 and 11:27 ISC for the exam. And the rest of you I'll see whenever you come by my office to pick up your exam. Go get some sleep.